Osmoregulation is controlled solute regulation via gain and loss of water across the plasma membrane between external and internal fluids. A substantial amount of marine animals are osmoconformers, whereby their internal fluids are isosmotic to their surrounding environments. Isosmotic physiological saline of the external environment is of relative pressure to the internal body fluids. To comprehend, let's compare the salmon and bull shark. Salmon belong to a group of anadromous fish. Spawning in fresh water, a leaven commenced multiplication, adaptation to salt water. The transition is made possible by their ability to osmoregulate. Gills contain molecular pumps and operate a countercurrent exchange system. Marine fish are hypoosmotic, diffusing water and salt ions via drinking and excreting salt ions and water through their gills, decreasing urine production. Freshwater fish are hyperosmotic, gaining water and salt ions, particularly sodium, through their gills while constantly losing salt ions and water through copious amounts of dilute urine. During transition, the molecular pumps are reversed, pumping sodium out rather than in. While residing in fresh water, sodium is pumped into their bloodstream via water flow through the gills. Salt water contains significantly higher sodium concentration, which means pumping the sodium from their bodies out into the salt water via gills. Cortisol triggers this hypoosmoregulation. The membranes of these chloride cells contain molecular machines called sodium-potassium adenosine triphosphatase, or NKA. NKA is the regulator of sodium and potassium levels within the cell membrane. Located in the epithelial cells within the gills is an enzyme that breaks down ATP, and the energy release moves both sodium and potassium ions against their concentration gradient. Once salmon reach sexual maturity, they migrate back to their place of birth to spawn. Once they successfully reproduce, their body slowly shuts down, resulting in mortality. Marine-dwelling elasmobranchs, inclusive of Carcharhinus lucus or bull shark, are urohaline elasmobranchs, capable of withstanding wide variances of salinity, often venturing to fresh water for long periods of time. This transition is achievable via physiological processes permitting them to independently control urea levels within the body fluid. The gills, kidney, liver, rectal gland and drinking processes are associated to regulation of urea, sodium, potassium levels and endocrine control to acclimatize to different salinity. Within saltwater environments, they are considered hyperosmotic regulators, whereby plasma osmolarities are significantly higher than freshwater environments, where they are considered hypoosmotic. In response to changing salinities of their environments, fully and partially urohaline elasmobranchs are capable of altering their kidney function and storing urea within a large liver. Urea storage results in an increase in ionic composition, preventing water loss. In saltwater, bull sharks possess higher levels of ionic compounds, more specifically, significantly higher levels of urea and TMAO, which protects important proteins from urea. When moving to freshwater, renal glands decrease urea levels, allowing selective adaptation between freshwater and saltwater without a transition period. Progressive increases in urine flow allows elasmobranchs to achieve reduction in sodium, chloride and urea plasma levels, allowing them to acclimatize to dilute environments. A larger reabsorptive capacity for urea via their kidneys allows acclimatation to opposing conditions. Varying urine output through kidney function is integral for bull sharks to control extracellular fluid volume contents to acclimatize to surrounding salinity variations. Rectal glands aid in eliminating excess amounts of sodium and chloride acquired from living in high salt conditions by increasing activity of the sodium-potassium ATPase ion pump. In order to maintain plasma ionic homeostasis to survive in differing salinities, rectal glands lower sodium-potassium ATPase activity, reducing salt excretion in fresh water. Osmoconformers do not regulate osmolarity and rely on their blood salt content levels to closely reflect their surroundings. This facilitates survival in varying salinities similar to their blood. The salmon and bull shark overcome amazing physiological challenges in order to transition between these salinities. Both practice osmoregulation, but the bull shark can also selectively ionoregulate, allowing easy and immediate transition to and fro.